Welcome back. In today's lab, we will be discussing fungi. So let's go ahead and get started. So what are fungi? We know that they're eukaryotic organisms, and there's actually three different types of fungi. That's going to be the yeast, the mold, and the mushrooms. In this class, we will be discussing the yeast and mold, and we're not going to get into the details on the mushroom. So they are a large group of eukaryotic organisms. And what's interesting about fungi is that they can be unicellular or multicellular organisms. Now, when you look at these pictures that I have here on this slide, can you guess which of this fungi are unicellular just by looking at them? Now, this is the way they appear, okay? This is the way that the mushrooms grow from the earth. This is the way that the mold would grow on an agar plate, or you may actually see it growing on some food items, for example, or on the wall if you have contamination or moisture. And this is the way that the yeast would appear under a wet mound. Can you guess which one of these is a unicellular eukaryotic organism? That's right, it's the yeast. You can see that they are small, unicellular fungi. In fact, yeast are unicellular facultative anaerobes. Ooh, what does facultative mean? Do you remember what that means? It can grow in the presence or absence of oxygen, right? And they actually can, depending on the yeast, they can form structures called pseudohyphae, which are false hyphae, false little filaments. And we're going to discuss a little bit more about pseudohyphae and hyphae in general in the lecture. Okay. Yeast also have many industrial fermentation processes and commercial properties, including production of wine, of bread, of beer, right? So these are small, unicellular, eukaryotic organisms. Moving on to the mushrooms. What do you think mushrooms are? Are they unicellular or multicellular? Well, the fact that you can see it, it has to be more than one cell, right? So these are multicellular organisms, as is mold. You can see the mold, right? So it's a multicellular organism, and you can see that there's various shades of it here, and there's growth in the medium, and then there's growth that protrudes out. So mushrooms and mold are both multicellular organisms. However, they do have some differences, you see. Mushrooms have organized multicellular filaments. They're organized. You see the beautiful structure. They're organized filaments. Mushrooms are also aerobic eukaryotic organisms, right? And we can see that they form these large bodies on the top that we refer to as fruiting bodies. And the mycelium, which is going to absorb this nutrients and stuff, will be below the ground actually. And now with the mold, another multicellular eukaryotic organism has non-organized filaments. In fact, you can see how fuzzy it is. It grows all over the place, right? Now those filaments are referred to as hyphae. Again, we'll get into the details of hyphae in the lecture. All right. And now when we have a group of these hyphae, we refer to them as mycelium, and that's going to be your visible mold that you see. These mold can sometimes actually produce some exoenzymes to help them digest food into smaller molecules, and that can be absorbed by the fungus. Now, there are some structures that you need to become familiar with when we're talking about mold, and so that's what we're going to discuss next. Imagine that this is the plate here, the one that you see of this picture of the mold. Here we see it on a plate and this little sort of cartoon picture. What we see is that the vegetative hyphae is what's growing within the media. It's embedded in the media. And this is the part of the mold that is actually absorbing nutrients. It's referred to as vegetative hyphae. Okay. Now, what we see growing on the top is going to be these little filaments or hyphae or aerial hyphae. So the aerial hyphae, also referred to as reproductive hyphae because it has the fertile hyphae there. You see those little spores. These are going to be spores that are going to be produced by the hyphae. And the hyphae usually extends away from the food source. All right? Away from the food source. Now, there are two different types of spore morphologies that we're going to get into some detail. But before we do that and continue our subject on mold, I have another picture here that depicts the vegetative hyphae being embedded within the food source that absorbs the nutrients, as you see here in the bottom of this picture on the right. And then what protrudes out is going to be your aerial hyphae, and that's the visible part. That is the visible part. So let's continue. There's a vegetative hyphae. So these are the two properties of a mold that you should know and understand: vegetative hyphae and aerial hyphae. So the aerial hyphae is going to contain the spores in the mold. So let's continue on with that topic of spore morphology. This is a nice picture because it shows you, well, you can kind of sort of not really see the vegetative hyphae being embedded within the media, but what you see protruding out is the aerial hyphae, and within that you see all these little spores. So there is something to be said about the morphology of the spore. All right, and there's going to be two different types of spores that you are responsible for knowing and understanding. And that is going to be a sporangiospore versus a conidiospore. All right. So what is what? Here we see, I like this figure, but I also put this one here for you to take a look at. We see the aerial hyphae is the stem that protrudes out from the vegetative hyphae that is being absorbed into the media to grab nutrients, right? So we see that this aerial hyphae is this filament that protrudes out. And then at the top, we'll see the various vesicles or the various conidiospores. So on the picture on the right here, what you see growing from the aerial hyphae is that this sporangiospore, what it is, it's a spore. The spores actually are enclosed within a sac. You see that? So these spores are all enclosed within the sac. Whereas in a conidiospore, that's not the case. The spores are out there. You see that? They're on the outside. They are not enclosed by a sac. So very different morphology in the spores. So let's go over the three molds that you are responsible for understanding and knowing in this class. That is going to be Aspergillus, Rhizopus, and Penicillium. So let's go over Aspergillus niger first. Okay, Aspergillus, as you can see here, well, it looks like a black mold, doesn't it? Yes, it is. It produces a black mold, and actually Aspergillus is a common contaminant of foods such as onions. So you may have seen an onion that stays too long in the fridge and all the moisture that builds up. You can actually start seeing some mold. Well, most likely that's Aspergillus niger, okay? Common contaminant of foods like onions, grapes, actually even peanuts. Yeah, but you know what's interesting is Aspergillus niger also has some important commercial properties in its production, well, its discovery of the production of citric acid. Remember what citric acid is? Yeah, it's an intermediate in the Krebs cycle. In fact, Aspergillus niger, production of citric acid from Aspergillus, is a multi-billion-dollar industry. Very, very interesting. Okay, so what do you need to know about Aspergillus? Well, I would ask you, what type of spore does it produce, right? So let's investigate. Let's take a look at the spore here. Here are two different pictures of a spore. This is under high power magnification. The reason that they're different colors is the different dyes that were used. But take a look at that. Look at its morphology and try to see if you can figure out, based on what we talked about in the previous slide, what are your options? Is it a sporangiospore? Well, what does sporangiospore mean? Spores enclosed within a sac. Or is it a conidiospore? What does that mean? A spore that's not enclosed by a sac. So we're going to get back to that in a second. So don't cheat, just try to see if you can guess, and then we'll go over the results. Next one is Rhizopus solanifer. Rhizopus is, it's also black, as you can see. Yeah, you see that? This is going to be your common black bread mold. It's probably actually the most common mold around the world, really. It's a very common mold. Okay, now that you know that when we're talking about mold, there are two parts, right? Or two structures. There is the vegetative hyphae that absorbs the nutrients, and then there's the aerial hyphae where the spores are produced, correct? So when you see mold growing in a bread, do you just pinch off that little area? What you're pinching off is the aerial hyphae, right? But what you may not see is the vegetative hyphae. Food for thought there. Okay. What does that look like under the microscope? Ooh, look at this one. Look at this beautiful cap here. And look at this one. It's bare. I wonder what happened there. And what is this? This is a mature Rhizopus zygospore. Okay. This means that two strains of the mold will fuse within the zygospore to produce many essentially diploid nuclei. All right. So this is how they reproduce essentially, but we're not going to get into the details of this in this class. Okay, so take a look at the morphology of the spore. There's differences here that we'll discuss in a little bit. I wonder what happened to this one, or what happened to this one, right? Okay, moving on to the next one. The third mold that we're going to talk about is Penicillium notatum. Penicillium, very, very interesting. You can sort of make out that it has a greenish color to this mold, I hope. I have other pictures that I'll show you that maybe shows it a little bit better. What does it look like? Here we see a little bit of green. Notice this sort of white outline when it grows on a plate. Under high power magnification, this is what the spores look like. This is actually under low power using our microscope, so you can kind of see it. Looks like a little branch, doesn't it? Very pretty. And this is what it looks like under high magnification. So you can take a look at that. Okay, look at the spores. What kind of spore do you think it forms? Now, going back to penicillium, we know that penicillium has commercial properties. Well, what is it? Penicillin, an antibiotic, right? Have you ever seen this sort of structure in any food item at home? Perhaps you've seen it in an orange, a moldy orange. That's penicillium. Now, don't try to take it if you're sick because it's not pure, right? You'll poison yourself probably or make yourself sick rather. So you don't want to do that. The one that you take from a pharmacy has been purified and so you don't want to do that, right? So this is penicillium that usually grows on citrus fruits if it's contaminated and old, right? Okay, so let's go back now. Aspergillus niger, what type of spore does it form? Did you figure it out? Conidiospore. That's right. The spores are not enclosed in a sac. Take a look. They're protruding out. Do you see them? You see that? Very, very nice. Okay, what about Rhizopus? What type of spore does it produce? A sporangiospore. Well, how so? What about this one? Ooh, very interesting question. You see here that the spores are enclosed within a sac, right? In this one, the spores have been released. The spores have been released. The sporangium has released its spores. And this is what remains when the spores have been released. Okay? It's this umbrella-shaped columella is what is referred to as. But you don't need to worry about the terminology for that. And then lastly, penicillium. What type of spore does penicillium produce? I think this is a little bit easier to tell than the aspergillus, do you think? It definitely doesn't look like the sporangiospore here. That is a conidiospore. Moving on to yeast. There are 3 different types of yeast that you are responsible for knowing for this class. At first glance, what strikes you? What do you think? Perhaps that the appearance of the yeast, the way that it grows, is similar to a typical bacterial colony and not fuzzy like mold colonies. Do you think? Yeah. If I gave you a plate of this, would you think that it is a fungi? Probably not. You would probably think that it's a bacterium. Very interesting. That a yeast, which is a fungi, would grow more like a typical bacterium than your typical mold. Huh? Yeah. So there are 3 different types of yeast that you are responsible for understanding: Saccharomyces cerevisiae, Candida albicans, and Rhodotorula rubra. Another thing that you may notice is that they have different pigmentations, right? They're very, very pretty. In fact, look how they grow when you do a 4-quadrant streak plate. You have isolated colonies, just like you would in bacteria. Okay, so let's investigate a little bit further. Saccharomyces cerevisiae has, as you may know, a lot of commercial properties. It is used for the production of beer, of wine, of bread, right? But notice one of the things that I want to highlight here. When we look at this under high magnification, we see that these are small unicellular microorganisms. And what's highlighted in red or circled in red is the way that yeast reproduce. Do you know what that's referred to as? Budding. That's right, budding. Okay? That's one of the characteristics of yeast. Yeah. They bud out. Okay, the next one is going to be Candida albicans. Candida albicans is the yeast that is commonly associated with yeast infections. Okay, so do we become contaminated or how does that happen? Actually, Candida albicans is normal flora. It's in women, in the vaginal area, it's normal flora, and it actually is in balance with bacteria that we have in that area as well. So then how do we get yeast infections? Perhaps we're on antibiotic treatment, kills the bacteria, and allows the candida to overgrow. Remember that microorganisms, when given the opportunity, can cause infection or disease, right? Yes. So that's one way that candida can overgrow. Not a problem. You can go take some over-the-counter medications and control the growth or overgrowth of Candida albicans. Another reason that women may be more prone to yeast infection could be douching. And so there's things like that that can cause pregnancy, the hormone changes that can all lead to an overgrowth of Candida albicans. So one of the ways that it can be determined that it is a yeast infection due to Candida albicans is the fact that Candida albicans forms what's referred to as a pseudohyphae. Look at that. Here is our own picture under high magnification. What's circled in red, you can see that production of the pseudohyphae. This is a characteristic of Candida albicans. What does pseudohyphae mean? It means a false hyphae. Remember that the hyphae are seen in mold. And what the hyphae is, it's a filament. It's that filament that grows out and holds the spores, right? The aerial hyphae? Yeah. Well, Candida albicans doesn't have that, but it forms what appears to be a hyphae. So that's why it's referred to as a pseudohyphae. Okay. And finally, here we have the yeast Rhodotorula rubra. Look at this color. It's beautiful. It's like a pink reddish color. These are moist looking colonies. All of them are sort of moist looking colonies if you really think about it. Rhodotorula rubra is one of your more common environmental yeasts. It is found in the soil. It can be found in the ocean, even in the air, and it produces these reddish pink colonies that are just really pretty. And it can be confusing because it could look like Micrococcus roseus. Ooh, look at that, right? Okay, so what does this look like under high power? Take a look at it. Remember, these are unicellular microorganisms, unicellular fungi. Does it resemble more like the Saccharomyces or the Candida albicans? What do you think? Do you see any pseudohyphae? I don't see any. Do you see budding? Yeah, I see budding as well. Okay. And so this will conclude today's lab.